Yeah, like Tom said, I'm Chantelle and I'm part of the Green Fuse group, which is Jim Weller's group. And today I'm just going to present a little bit of work that I've done in my PhD as part of the group. But first, I too would like to acknowledge the traditional owners and custodians of the lands which we meet on today and pay my respects to their elders past and present. So our group's really interested in domestication. So that's where wild species have been adapted for human use and cultivation through selection. And this has led to distinct phenotypic changes. So this can be things that are really critical for domestication, like reduced seed dispersal or reduced dormancy. But depending on the species, it can also be other things like earlier flowering or reduced branching. And having knowledge about the underlying genetics of these changes can be really useful for a number of reasons. It can tell us about what genes control a trait, but also about the history, so where and when these changes occurred. And finally, it's also important practically. So as people start to seek to incorporate genetic diversity from wild relatives, it's important to know how they actually differ. But our understanding has mostly been focusing on species like barley, maize, and rice. And despite the importance of crop legumes, it's surprising sort of comparatively how little we know about them. So our group's interested in legumes. We focus on legumes, and there's loads and loads of legumes that are important crops globally. In the last sort of 5 to 10 years, there's been really significant progress being made in soybean. But compared to sort of these other legumes, we know surprisingly very little. So that being the case, there's still a bunch of interesting questions that we can ask in terms of the genetics behind traits of various legumes here. And things like environmental adaptations and going all the way from domestication to their diversification. So I've been focusing on these two legumes in particular, so common bean and pea, and they're kind of in this like really sweet spot where they're between the kind of model legumes like medicago, and they're also between kind of crop legumes that we know a lot about like soybeans. So they're kind of in this nice area where we know enough about them to start making some of these questions answerable, but we still have a lot to find out. And what's important specifically to the domestication of these different legumes is a little bit context dependent, but again, we have similar suite of traits being selected for, like reduced dispersal and reduced dormancy, but also different changes to plant architecture as well, and seed size. So today I'm just going to kind of focus on a couple of traits as illustrations of the approach that we've been taking to understand the genetics behind legume domestication. So I'll cover some work that we've been doing on seed weight in bean and touch briefly upon some dormancy stuff, and then also the work that we've been doing in pea for flowering. So broadly, we've been taking this biparental QTL analysis approach, which just kind of allows us to identify chromosomal regions that are influencing traits of interest. And it's, of course, both of these are legumes, but they also share a lot of differences. So they're quite phylogenetically distinct. So by using both of them, we're kind of able to tackle different parts of that overarching question of domestication as well. So common bean is actually one of 5 domesticated species in the Phaseolus genus. And it's got huge diversity, and it's a really interesting crop to look at for this question of domestication, because it's actually undergone 2 independent domestications around 8,000 years ago in the Mesoamerica and in the Andes. And this has resulted in 2 major gene pools which contain both wild and domesticated forms. And there's been lots of great work in the common in the common bean space. So including work from our group here, focusing on flowering time adaptation, but other groups also identifying genes which confer determinancy and pod shattering. And it's kind of a nice example as well of, if we look through the genetic diversity, we're able to say that, you know, these genes have been independently modified in these two gene pools and also acquired in different orders. So we know a little bit about domestication in common bean, but there's still a lot of aspects that we don't really know anything about the genetics behind. So things to do with other aspects of plant architecture like elongation or branching, but also seed size. So again, that common approach that we're taking is making a cross between a wild and domesticated bean. They're varying in seed size quite dramatically. Create the F1, self that, and then in the F2, that's where we measured a bunch of different traits, genotyped them for molecular markers, and constructed a genetic linkage map. That then allows us to perform QTL analysis, and as is the way when you measure a bunch of traits, you get loads and loads of data, and so you're often able to identify loads and loads of QTL. So each one of these sort of colourful dashes is a QTL that we've been able to identify. Then the challenge from that really is, how do you make sense of that all? So today I'm just going to focus on the seed size QTL that we identified in this population. So we identified 11 seed weight QTL on 9 of the 11 bean chromosomes, and 4 of those had a PVE, or a percentage of variation explained, of over 5%. So combined, these 4 QTL explain about 30% of the variation that we observed in seed weight, and the strongest of which on chromosome 7 here was a bit interesting in that it also co-located with several QTL for things like leaf size and early elongation as well. And like with all of our seed weight QTL, we found that the domestic allele conferred increased seed weight, which is probably to be expected. So this is great. So we've got a region of the genome that we know is influencing seed weight. But this region here is still quite large. You know, it contains hundreds of genes in there. And so one approach we've taken is this idea of fine mapping. Basically, what you do is you generate an F3 population that's segregating at your QTL region, but fixed for other regions that you think are influencing your trait of interest. You can make molecular markers, and you can essentially track where different regions, from what parent different genomic regions come from. And then you can test for sort of statistical significance between that marker and the trait. So if you're getting sort of part of the genome from the parent that has the causal kind of mutation for increased seed size, you can hopefully see that segregating out and see a phenotypic change associated with that. So this can be a lot of work to sort of grow and generate these subsequent progenies and populations, but it's really worth it to validate that QTL region and also narrow the number of genes down as well. So if we look back at that chromosome 7 QTL, here this bar just sort of represents that QTL region. So it was about 345 genes are in that region there. And what we did is we populated it with a bunch more molecular markers, so to be able to track which parent that genomic region came from. And this one in the red here is just where our original QTL peak marker was. So we grew that F3 population that was segregating across that QTL region, and great, we saw, we could still pick up a significant effect of that locus. So that sort of just validates that the QTL is real. It's not just some figment of statistical association, which is always good. But that region is still quite large. So what we did is through working with this population, we identified a number of lines that had interesting recombinations. And this essentially allows us to break this big chunk up into 3 separate smaller chunks. And basically we can see, is our locus located in any one of these chunks by seeing if we can still pick up the effect of that locus. So if we look at population 2 first, that's just segregating on this right-hand side, we don't pick up any effect of our locus here. So effectively, we can sort of exclude this right-hand side of our QTL region. If we look at our next population, population 3, that's segregating on this left-hand side, we see the exact same thing. We don't pick up any significant association. So again, we can exclude the left-hand side of our region. So we've already kind of narrowed it down to this region in the middle. And we then grew population 4, which will just hopefully pick up the effect to confirm that it's in that region. And that's exactly what happened. So something segregating in this yellow region here is where our locus is. That's where we're picking up this significant effect still. So that's really great. So we've been able to sort of narrow it from 345 genes down to just 47 genes. And sort of in that region, we have a couple of interesting candidates, but by far the most interesting one is this divergent CCT motif. And that's because if we look at sort of the other literature in legumes, in soybean, they had an induced mutant for this gene called big seeds 1. And if you blast big seeds 1 to the common bean genome, you find that it's actually this gene here, the divergent CCT motif. So in soybean, the mutants have increased seed weight, but they also have increased leaf size. Natural variation at this locus has also been found in chickpea to do the exact same thing, as well as increase hypocotyl length. And we see the exact same suite of phenotypes in Arabidopsis as well. And if you might remember, we also saw co-location of QTL for leaf size and early elongation as well. So that's just like an extra little bit of supporting evidence. And we can pick that up in subsequent populations as well. So this is where we are at the moment with this QTL. And it's kind of a nice example of how we've tried to follow up other QTL regions that we've identified in common bean as well. Just have a drink of water, sorry. So as I said in the beginning, we're also working with pea. And we've taken a similar approach as well with that QTL analysis and fine mapping. But we also spent a little time looking at trait diversity in the genus as well. So pea is one of the oldest domesticated species, around 10,000 years ago. And it's been domesticated in a different part of the world compared to bean. And the genus is a little bit messy. It's sort of best described as a species complex. And we can see sort of in the genus, there's, a number of distinct genetic subclades as well. So the original distribution of, we generally consider there to be 2 species, Pisum eladius and Pisum fulvum. If we take Pisum eladius, the sort of original distribution is quite broad, and it's sort of all of that grey that's left here. But we also have this second species, fulvum, which has these beautiful distinct yellow flowers, and is also more genetically defined and has a more restricted geographic distribution as well. And sort of from this broad eladius complex, we consider there to be the domesticated peas to have arisen from there. So in the blue, we have Pisum sativum, which is the pea we all know and love, the garden pea that you guys will all be familiar with. And that subsequently spread to the whole world. But we also have this second, less known domesticate Pisum abyssinicum, which is sort of nestled between these 2 eladius groups here. And it has this really restricted distribution to parts of Ethiopia and Yemen, and is really quite genetically distinct. And as you can see, there's no sort of wild peas currently found where abyssinicum lives. It's also important to note that there is some incompatibility issues, but there's not a complete barrier between these groups. So it's really cool. Again, we have these kind of two domestications. They don't appear to be close genetically, but they do have a similar suite of those domestication traits. So they both have reduced pod shattering, reduced seed dispersal. Abyssinicum also has some distinct characteristics. So the serrated leaflets and often sort of black glossy seeds. And it has, again, that really restricted distribution. So first, what we did is we were kind of looking, or interested at looking kind of at the genus level of trait diversity, as there's not really a lot of information widely available for this. So we're interested in like what traits might distinguish these groups, what might be different between them. And to do that, we sort of constructed this mini diversity panel. So about 90 accessions that span all the major groups of P and vary dramatically for various different traits. So we then measured a bunch of phenotypes, too many to show today. Just as an example, here's some data for branching under short days. So as you can see that we have huge sort of variability across the genus in branching. And we can look at the data we've collected sort of by accession like this, but we can also summarize them in terms of groupings. So for instance, we've got days to germination on the left here. We nicked a bunch of seeds, and then this is how long they took to germinate after we nicked them. So we've got some example of sort of physiological dormancy here as well, which we weren't really expecting. We also have the trait that we love to focus on in this group, flowering time, and we can see that there's a huge range in the domesticate sativum. Some accessions flower early, some flower late. A lot of the wild tends to flower late. We have the wild fulvum, which has the capability to flower a little early. This really weird Aladius group that seems to have a couple that flower really early. And we have Abyssinicum here that's always flowering really early. And that makes sense when you consider kind of where it's grown in Ethiopia, where it's at those lower latitudes. So it kind of needs to flower early under these non-inductive days. So it's great that we had a look at that trait diversity, but we're really interested in getting down to the genetic level of these traits. So one question we might ask ourselves is, since we see a lot of similar traits between Abyssinicum and sativum, we might ask, where did those traits come from? You know, is it the same genes that have been modified in sativum? And if it is the same genes, is it the same mutation? We might imagine that there could be some sort of integration from the other domesticate, or it could be completely independent, kind of like how we see in bean. So we're interested in asking, is there conservation at the genetic level for domestication traits? And to try and tackle parts of this question, again, we use that approach of a QTL analysis, fine mapping in an F2 population, this time between Abyssinicum and a wild Aladius. And I'll briefly just touch upon some of our dormancy results, but really we've been focusing on flowering due to that importance of where Abyssinicum grows. So in terms of dormancy, we identified 5 QTL in Abyssinicum. That seems to reduce dormancy. And if we compare that to earlier work in our group done on sativum, we see that we have about 3 dormancy QTL that appear to co-locate with sativum. So this is suggestive that it might be the same loci being selected for dormancy. But really, we've primarily been focusing on flowering. So in Abyssinicum, we identified 4 flowering QTL. The strongest 3 had candidates all from the same gene family, quite strong candidates. So 2 of which were phloragen genes, which promote flowering, and 1 is an anti-phloragen gene, which represses flowering. And we found in all cases that the domesticated Abyssinicum allele confers earlier flowering, which is what we would expect. So this suggests that this is probably a gain of function and this is probably a loss of function. And again, we find that QTL that other work in the group identified for sativum co-locates with our QTL for Abyssinicum. So again, suggesting it's likely the same loci that have been selected for. And so one question we need to ask is, was this independent or was this introgressed? And one way to examine this is to sequence these candidate genes across the major groups using that panel that we constructed, and then seeing if Abyssinicum shows any affinity with sativum or with any other particular groups. So really we're asking the question, are Abyssinicum and sativum sequences the most similar? And if they are, that suggests that there might be some integration. So we managed to obtain partial sort of sequence for each of our 3 flowering candidates across that diversity panel that we created. This was a little bit difficult because the material was so diverse, so we did run into a lot of difficulties here. So we didn't obtain sort of huge amounts of SNP variations, but enough to give us an idea about affinities. So the first 2 genes weren't particularly clear. We sort of have Abyssinicum clustering with a group of sativum and an eladius individual for FTA3. For TFL, we have Abyssinicum clustering pretty confidently with eladius. And with FTA1, we had a really surprising result where it seems pretty definitively to be clustering with fulvum. Suggestive this region might have come from fulvum or a common ancestor of this group, which we weren't expecting at all. So overall, these candidates show different affinities, but integration from sativum seems unlikely. So it seems to support, yes, these are 2 independent domestications, and the way that they acquired these domestication traits appears to be independent. So that was just some of the work that I've been doing in my PhD. Broadly speaking, we're hoping to increase our understanding about the genetics that are contributing to domestication traits in both bean and pea. We've convinced ourselves that this is a sort of interesting and valuable approach of using this QTL and fine mapping framework, and that there appears to be sort of value in pushing ahead for traits in the quest to find causal genes as well. And interestingly, we're seeing quite a lot of loci for a number of different traits that are conserved across legumes. And of course, it's important to note this is all preliminary. It would be great to have more sequences, more fine mapping, definitively prove causality, but this will, of course, take more time and work. So yeah, thank you very much for listening, and if there's any questions.